Hello, good morning, good afternoon, and good evening to all, depending on the time zone that you're in. My name is Patrick Limsu, and I'm currently a Senior Principal Scientist at Pfizer and co-leading the Nanomaterial and Nanotechnology Working Group with my co-chair, Elaine Pugh, Associate Scientific Director at Bristol Myers Squibb. We are pleased to present to you the first IQ webinar hosted by the Nanomaterial and Nanotechnology Working Group, which is part of the Drug Product Leadership Group in the IQ Consortium. Our speaker today is Dr. Jeffrey D. Clarkson, who is a principal scientist and head of the Physicochemical Characterization section at the Nanotechnology Characterization Laboratory, NCL, which is part of the National Cancer Institute. Dr. Clarkson is an expert in physicochemical characterization and standardization of nanoparticles. He leads a team that develops new analytical methodology for critical quality attributes and assesses current and upcoming instrumentation for nanoparticle characterization. He received his PhD in chemical engineering from The Ohio State University. Dr. Clarkson has published a number of publications and reviews in the areas of physicochemical characterization of and in vitro release from lipid-based drug delivery systems and analytical methodology. As you can see from the title, the title of the presentation is Nanomedicine Development, Physicochemical Characterization Considerations. Dr. Clarkson, the stage is yours.
Thank you, Patrick. Can you hear me and see my slides before I go on?
Yes.
Yes, okay, perfect. So thank you, Patrick, for the warm introduction. And thank you to the organizers for allowing me this opportunity to speak with you today. As Patrick had stated, the title of my talk is Nanomedicine Development, Physicochemical Characterization Considerations. Before I go into that, though, I do want to spend a couple of slides on the NCL, or Nanotechnology Characterization Lab for short, namely who we are and what we do. So the NCL was established back in 2004 as a collaboration between NCI, NIST, and FDA with 4 primary objectives. The first is to characterize nanoparticles using standardized methods, to conduct structure-activity relationship studies, to facilitate regulatory review of nanotech constructs, and finally to engage in educational and knowledge-sharing efforts like I'm doing today. So we work with the extramural nanotech community. So typically these are academia, small and large pharma, other government agencies, and they submit their particles to us via a white paper process. And this information can be found on our webpage here. Once it is accepted to the NCL, their nanoparticles are subjected to an assay cascade, which is consisted of physicochemical characterization, immunology, pharmacology, and toxicology. We work with FDA and NIST, namely for insight and guidance in terms of characterization. And finally, the characterization is used for our client in terms of their preclinical development, clinical translation, regulatory, and hopefully commercialization needs. Because we are developing typically new methodologies for characterization of these nanoparticles, we work with ASTM, USP, and ISO. And namely, that is to get our methods out there as methodology guidance documents or SOPs. We also have within NCL a small formulation group. And the ideal here is that that group can help formulate your product or reformulate existing drug products. We have over 15 plus years of knowledge, experience in this area. And our main mission here is to help accelerate the translation of promising nanotech drugs and diagnostics. And like I mentioned, everything can be found on our webpage. If you take anything from this slide, it is that this assay cascade is a free service. This assay cascade consists of 3 different sections: physicochemical characterization, in vitro, and in vivo. And what I've done here is listed basically the type of experiments that we do for each of these different sections. I'm not going to go through this, but the main point here is really to show that NCL testing links the physicochemical properties to a biological outcome. And a lot of our assays here, the protocols that is found on our website given below. So just in summary for NCL, we do support preclinical characterization. We do help with regulatory concerns, clinical characterization. We do explore alternate indications as well as next generation nanoparticles. We currently have 17 collaborators in clinical trials with novel nanomedicine therapies, and their logos are shown here. If you want further information on how about to apply to NCL for characterization, it can be found on our website here. With that, I want to now switch over to the bulk of my talk, which is really looking at the physicochemical characterization. So when you think about physicochemical characterization, or PCC for short, it really boils down to the analytical instrumentation and development of new methodologies. So what I have listed here is all the different techniques that we have in-house at NCL. So we have a lot of light scattering techniques, DLS, MALS, laser diffraction. We have microscopy, both TEM, AFM to look at size measurements. We do have resistive pulse sensing as well to look at size and size concentration. We do zeta potential. We have a whole suite of chromatography separation systems, as well as CHNOS elemental analysis, spectroscopy, and thermoanalysis and QCMD. And really what I want to do here was try to basically take this information that we have and leveraging over 15 years of experience, we have identified the critical quality attributes and the methodology needed to support the most common nanoparticle platforms, these being liposomal products, polymeric nanoparticles, and colloidal metal nanoparticles. Now we have all this knowledge here, and trying to get that or share that with the community, this information is kind of hard. I always say, it depends on the particle. Every particle is unique, and that applies to its characterization. So what I did, in an effort to try to share all the knowledge that we have at the NCL, I looked at guidance from the FDA, namely this liposome drug products guidance for industry. If you look at this, excuse me, sorry about that. At this guidance document, contained within it is a CMC section, or your chemistry, manufacturing, and controls. And within this subsection, you have critical quality attributes. So what I did was basically go into this document and fish out all the critical quality attributes needed to characterize liposomal drug products. And I basically distill this information into what I call the parameters, methods, and considerations for the PCC of liposomal products. If we look at the CQAs needed, or that should be assessed for these products, we have size distribution. And what I've given here are examples of the type of techniques one can use to assess size and size distribution. So we have our DLS, which pretty much everyone does nowadays. We have MALS, cryo-TM, and AF4, for example. We then have morphology. Typically, cryo-TM is used to evaluate morphology for liposomes, and this gives you information on size distribution, liposome morphology, things like that. And then surface characteristics. So this refers to the surface in regards to zeta potential, for example. We do protein binding assessment with AF4, and we can also do QCMD to look at binding constants. We then have composition as a CQA. Now, in the previous 3 CQAs, I gave you techniques that one can use. For composition and the remaining CQAs that I will discuss, I don't give you the techniques because it really depends on the nanoparticle, but rather what I've given you here are the types of questions one should consider when characterizing or assessing that CQA. So for example, composition, we have drug concentration, so total, free, encapsulated. We have drug distribution as a function of size. If you have any targeting, you need to know how much you have on there, both total, bound, and unbound. With liposomes, it's good to look at the individual lipid concentrations. For the case of certain drugs, we're using counter ions to precipitate the drug within the interior. It's important to know these interior and exterior concentrations, excipient concentrations, and so forth. We have purity as a CQA. So purity here is defined as drug and lipid impurities. This would also mean to any free drug, lipid, or targeted ligand concentrations, or any residual solvents or reagents due to the manufacturing process. Stability. So stability can be defined in many ways. For liposomal products, we can define stability in terms of size distribution. Does it aggregate with time? We could look at drug leakage and degradation. We have hydrolysis of lipids, drug release in plasma, things like that. And all of this can be used to assess storage conditions and shelf life. Finally, we have batch-to-batch consistency. So this is obviously very important. So if you make a batch today and then someone else makes it next week, they need to be the same. And you need to assess this by choosing the relevant parameters to define these release criteria. And they need to relate to a desired in vivo outcome. So a lot of times people will use size, for example, because it's probably the easiest thing to measure by DLS. But sometimes DLS is not enough or not a good CQA to assess batch-to-batch consistency. And you need to define or learn what that relevant parameter is to define your batch-to-batch consistency. And finally, we have starting material characterization. So a lot of times this is overlooked. People just assume when they buy something or pull something off the shelf, the material is good to go. And they wonder sometimes why their final product is a mess. Well, if you start off with bad or messy starting material, good chances is your final product will be a mess too. So you do want to characterize your starting material. So this would be in regards to the drug, the structure, identity, the purity. If you're working with lipids, you want to look at the lipid composition, the structure, the fatty acid distribution, and as well as purity, and also to assess the storage life of each of the individual starting materials. So taken as a whole, we want to use this basically as what I refer to as a cheat sheet to help with the characterization of liposomal products. And what I've done is basically put this on our website as a downloadable handout, and the link is shown here. So this is for liposomal products. I also extended this to polymeric nanoparticles. I'm not going to go through this, but same CQAs, slightly different methodology and questions one needs to ask for polymeric nanoparticles. And again, I have this available on our website down here below. And I did the same thing for colloidal metal nanoparticles as well, again, found on our website. So I urge you to check out our website. There's a lot of information. If anything, download these 3 files for your reference and for guidance. So the rest of my talk, I do want to share now with you some interesting methodology that we've developed here at NCL for characterizing not only liposomal products, but everything out there. At NCL, we pretty much put our hands on everything these days, so it's a good thing. And I just want to share some of the more interesting results with you. So first with lipid composition. So our client came to us with lipid nanoparticles with siRNA loaded, and we wanted to assess batch-to-batch consistency. So for this, we developed an HPLC method with charge aerosol detector, or CAD, and we came up with an elution profile that can actually separate out the 6 different lipids. And there's no, unique special properties for detection, so we had to use CAD in this sense. And with 2 runs, we can actually quantitate the 6 lipids in our formulation and use this to assess batch-to-batch consistency. The next example is looking at ion quantitation in liposomal products. And for this, I'm using Doxil as an example. So everyone's probably familiar with Doxil. When you load doxorubicin, you actually precipitate it out within the interior as a crystal. And this is what you typically see at a cryo-TM micrograph of Doxil. So we wanted to quantitate the amount of ammonium and sulfate in the formulation. So we came up with a reverse phase HPLC with CAD detection to quantitate ammonium as well as sulfate. These are the total amounts. And then using microcon centrifugal devices, you can actually measure the external amount of ions. And then through mass balance, you can calculate the internal concentration. This methodology does support FDA regulatory guidance for doxorubicin hydrochloride liposomal injection products. We use this a lot to compare batch-to-batches as well as to assess Doxil analogs. And then this method can also be extended for other counterions as well as excipients or buffer ingredients. And we published this in the paper shown here, which basically details this methodology. Still looking at Doxil, we want to look at impurities and stability assessment. So Doxil contains HSPC, cholesterol, and lipid PEG. When these degrade with time, they basically give you the free fatty acid, so stearic acid. If you're looking at DSPC, we have palmitic acid at the PSPC. And we wanted to see if we can track the presence of these free fatty acids as a sign of stability of these liposomal products. And for that, again, we came up with an HPLC with CAD detection method to look at the free fatty acids. In this case, we're looking at 4 batches of Doxil, each with different expiration dates, as well as the Doxil analogs made by Sun Pharma. And you can see the stearic acid peak here. And as expected, the older lot has more stearic acid, the more recent has less. So you can use this methodology to track the free fatty acid. You can also use this to track the corresponding lipo-PCs. So that's a good mass balance check. So it's a good way to assess stability and shelf life for these liposomal products. Let me look at now drug loading for a prodrug. So those of you who work with prodrugs, simply you're taking a drug and you conjugate this to a polymer backbone, typically through a cleavable linkage. And the question here is, how do we determine the drug loading for these prodrugs? It's obviously a CQA for prodrugs, and it's actually very hard to do. So traditionally what you do is you want to do some kind of hydrolysis method for this, because typically these drugs, when you conjugate to the polymer backbone, you might shift the optical properties or spectral properties of the drug, so you can't use UV vis anymore. Plus, typically these drugs are loaded at such a low amount that UV vis detection is just not sensible enough. So we need to come up with a chemical method, and hydrolysis method, like I mentioned, is the easiest, whereby you simply just incubate your sample in either acid or base, let it cleave, and then develop a reverse phase HPLC method to look at the free drug. Now, this takes a lot of time to develop this method. And the question always when you do this approach is how do you know you're cleaving 100% of the drug? You can't answer that. You just don't know. Moreover, what can happen here is as you incubate with longer times, you can start breaking any ester linkages contained within the drug. So it makes looking at the intact drug very hard and challenging. So we came up with a new method whereby we're using a very old technique. It's an elemental analyzer. Essentially, you're combusting the sample and you're looking at the carbon, hydrogen, nitrogen, and sulfur content of your sample. And we tested this on our in-house prodrug. So we have a prodrug with this is the backbone, this is the drug, and here's a prodrug with the conjugated drug to the backbone. We ran this through our CHNOS analyzer, and we looked at the sulfur content as well as the nitrogen content. In this example, our drug contains sulfur, so we can use sulfur content alone to assess drug loading. If you're working with a drug that doesn't have sulfur, you can always use nitrogen. But in this case, you need to run all the precursors. So in this case, you'd have to run the starting polymer backbone and the free drug by itself, along with the actual prodrug. And then through mass balance, you can basically calculate the weight percent of drug loading in your sample. So we did that with this, and we compared this with traditional methods such as hydrolysis and reverse phase HPLC, as well as size exclusion chromatography. And you see we're getting very good results across the board using very different techniques. What's nice about this technique is that it's very easy to do. There's no method development. You simply combust the sample. It's a very fast run, roughly 5 minutes per sample. You need about 2 mg of sample in a powder form for this to work. And it's very good. We've tested this on multiple samples. It's very good, consistent, and it's validated by other methods. And we published this actually in the paper shown here. So it's a new method to look at polymer-bound drugs. Using the same rationale, we also want to look at drug release of prodrugs in human plasma. This is especially important if you're working with prodrugs. Not only do you need to know how much drug is loaded on the prodrug, you want to study the release of the drug from the prodrug, typically in human plasma. This will mimic what happens in the body. So in this case, we're working with a prodrug that's loaded with in micellar form. Here's a cartoon representation of it. We want to ask questions of how does it cleave, where does it cleave, how fast does it cleave, and what's the stability of the drug after cleavage. So we came up with an HPLC method here, where instead of looking at the cleaved drug, we actually looked at the intactness of the prodrug itself as a function of time. So at time naught, we have a peak here that corresponds to the prodrug intact. As you incubate this with plasma, with time, this drug will be cleaved, and this prodrug peak will decrease with time. You can then convert this to your hydrolysis rate of your prodrug. The reason we did this, and I kind of mentioned this on the previous slide, is that especially with the longer time points, if you have any ester linkages in your drug, the esterases in plasma will start to degrade or break those bonds as well. And essentially you have a nice mess to try to analyze because that cleaved drug is no longer intact. It actually itself has cleaved itself. So this is another way or a different approach of looking at hydrolysis rates of prodrugs. We also did LCMS to separate out the components of formulation from plasma, and we can use LCMS to determine the cleavage flight as well as any drug epimers and cleaved drug products. Switching over to metallic nanoparticles, we want to look at protein coding. This is obviously important to know how much protein you have loaded on a metal oxide in this case. For this, we're using thermogravimetric analysis, or TGA. For those of you who are not familiar with TGA, essentially I will say TGA is basically taking a balance and then putting it in an oven. Turn the oven on, and you're basically burning off the organic, and the weight will decrease as a function of temperature. So we were looking in this case of a metal oxide with protein coating. We increase the temperature, we burn off this protein coating, and this will give us that weight loss corresponds to how much protein is on the sample. And in this case, since you have a metal oxide, you don't burn this off at the end. So that final weight at the end of the run will give you an idea of how much metal oxide you have in the sample. This is useful if you don't have ICP-MS, for example. And a single TGA run gives you both protein and metal oxide concentrations. Looking at another example with Go nanoparticles, in this case with protein and lipids, we want to quantitate the amount of protein and lipid coating on this. So for this, we're simply using KCN. KCN will dissolve colloidal gold, thereby liberating the protein and the lipids that were on the coating before. And then simply you can use fluorescence to quantitate the amount of fluorescence. And we can use reverse phase HPLC with CAD detection to quantitate the amount of lipids. So it's a nice little method to look at coating quantitation on colloidal gold nanoparticles. Let me switch over now to therapeutic nucleic acid-based nanomedicines. Obviously, this is important and in the limelight due to the COVID vaccines. So we wanted to look at miRNA quantitation. So we had a formulation from one of our clients where they submitted to us a polymeric nanoparticle containing 2 different miRNAs, and we want to quantitate total and free mRNA in a formulation. For this, we use traditional page, SES page for this, and you can see from here, the gels, you can separate out the 2 different miRNAs, and you can quantitate both and add them up to get the total. Now, this gives us total mRNA. It doesn't tell us anything about how much is free. So for that, we came up with a method where we're using actually a commercially available fluorescent assay kit. It's a qubit microRNA kit. It's based on fluorescence, and it simply reacts with any free mRNA present in the sample. So to look at unencapsulated, you simply just take your sample in water and run it through the assay, and this will give you any free unencapsulated mRNA. We worked up conditions where if we use HFIP, hexafluoroisopropanol, or triethylamine and methanol, this will essentially dissolve the polymeric micelle, liberate the mRNA, and then run it through the fluorescent assay kit. This will give us a total mRNA. And if we compare the results here, it's very good agreement between the traditional SDS page with this qubit fluorescent assay in determining how much total as well as free mRNA. So it's just another tool in our kit to look at, mRNA quantitation, both total and free. We wanted to extend this now to mRNA. Like I mentioned, mRNA is used in the COVID-19 in your Pfizer and Moderna formulations. We want to see if we can use this with mRNA now. We had a sample, again, a polymeric micelle loaded with mRNA. We want to see if we extend this, and actually it does work. Again, using the same type of condition shown here, we can look at unencapsulated as well as total mRNA in this formulation. Let me now switch over to a technique that's basically emerging as a very good technique to look at fractionation of samples and hence its characterization on a fractionated sample. And this is asymmetric flow field flow fractionation, or AF4. I have a cartoon of the side view of it. Basically, it's a channel with a semi-permeable membrane on the bottom, typically a 10K, and you have a channel flow, and your sample comes through, and you basically affect a cross flow, which is your driving force, which will pin down the larger particles to this membrane and allowing the smaller particles to elute first. So your elution order is small to large. There is no stationary phase in this case, as opposed to, let's say, a side solution chromatography where you have basically a column where your particle can interact. In this case, you have just a membrane. We can change the channel flow, the spacer, the type of membrane, and the cross flow to affect separation. Here's a picture of it in our lab. So here we have our AF4 channel in the middle. We have hooked up to this our UV. We have a Mols detector, RI, and inline DLS, and that's shown down here. We separate with AF4. We use inline DLS to measure hydrodynamic size, inline Mols to look at radius of gyration, and finally we do a lot of offline collection coming off the AF4 and do reverse phase HPLC on those fractions to measure drug concentration. So let me share with you some examples of how we use AF4 in our lab. The first one has to do with batch-to-batch consistency. These are 3 lots of pegylated metal oxide nanoparticles. We did TEM on them. Typically, when you have metal oxides, you do TEM. So the TEM micrographs are shown here. Now, if I asked you, can you tell me which is different from each other? You probably can't. They all look the same. And if we went to sizing these nanoparticles by TM, you can see that they're pretty much the same. So no difference by TM. So in this case, TM is not your best method to look at batch-to-batch consistency. However, if we use AF for mols, we can see that actually lot 1 is different from lot 2 and 3. So in this case, AF for mols is better suited to look at batch-to-batch consistency. We use a lot of AF4 to look at size and drug distribution and in human plasma. So this is an example. We have docetaxel and doxorubicin loaded liposomes. If we do a batch mode DOS, we get a pretty polydispersed sample or size distribution. So this begs the question where is the drug, right? You can pick any of these sizes, tell me what size has what drug and which drug, since there are 2 in this case. So we're using AF4 to separate out the nanoparticle with inline DLS, and then we're collecting fractions off of the AF4 and doing reverse phase HPLC for drug quantitation. So these are the results for the flow mode DLS for doxorubicin. So basically, the intensity of the light scattering is a function of time. And what we've done here is with the DLS sizes are given here, shown in green. We can see we have varying different sizes or size populations in the sample. And by collecting each of these fractions, we can quantitate the amount of drug associated with that size fraction. So for example, you see most of the drug is contained in the size between 35 to 80 nanometers. I should also point out that AF4 in this case can separate out the nanoparticle from the free plasma proteins. What's interesting here though is we're only seeing 62% of the total drug recovered, with about 2% free, and this is done offline with another type of analysis. If we look at the other drug, docetaxel, in this case, we're only seeing 8% total recovered across all these peaks. And the reason being is that if you're familiar with AF4, you typically passivate the membrane with bovine serum albumin, a protein, before you start running your actual sample. In this case, the docetaxel partitions to the BSA, so it has more of affinity for the protein rather than nanoparticle. And this can be used to basically assess drug stability. So if you think about it, if you inject this into your body, most likely the drug will be released to the plasma proteins. And this method kind of gives you an idea of how much it releases to the proteins. And we published this in this paper shown here. So it's a method to assess drug stability. What's nice about this is with 2 runs, you can do with and without protein or plasma protein incubation. You get size distribution of your formulation. You get drug distribution as a function of size. And finally, you get possibly drug stability of your formulation. So making AF4 a very nice method to use in characterizing nanoparticles. Here's another example, and this time we're using a polymeric micelle. If we look at the batch mode DOS, it's actually more of a mess than the previous example. Again, where is the drug located? We're using AF4 DLS with offline reverse phase HPLC. In this case, you can see AF4 can separate out the 2 populations with 2 different size ranges of a constant 55 nanometers and an increasing size population. In this case, we're only seeing 14% of the total drug recovered only. Again, this implies that a lot of this drug is partitioning to the protein. We follow this up with another technique that my colleague in our lab does, and it's based off the Sichuan method, and the reference is given down below here. But basically, they're seeing the same result where 85 to 100% of the drug is released when incubated with plasma. So you're getting very good agreement between 2 very different techniques to assess drug stability and release. We can also look at protein binding. Like I mentioned, you can run AF4 with and without plasma proteins, and AF4 can separate free plasma proteins from your sample. So in this case, we're doing AF4 with mols and DLS in line. And for this, we're going to look at 3 commercially available liposomes, doxyl, onivide, and ambisome. And I've also given you the lipid composition for each of these 3. The main thing here is to look at the lipid peg. So in doxyl, we have about 5%. In onivide, 0.3, and ambisome has no peg at all. We want to take each of these samples now and run it as is through our AF4 and then after plasma incubation. And the results are shown here. Let me walk you through this. The first panel is your AF4 mols. In blue is without human plasma, and in red is with human plasma. So you can see here the free plasma proteins, and here's our liposome peak. This is the DLS trace. So from both of these, you can calculate this shape factor defined as Rg over Rh, and you can calculate this before and after plasma incubation. In this case, they're roughly the same. So this implies minimal protein binding. If we look at Onivide, we conduct the same type of experiment, and we calculate this shape factor again. We can see there's a slight increase, about 12% upon incubation with human plasma. So this suggests there might be some protein binding to the surface. And as you can probably expect with ambisome, if we look at the shape factor here, it's almost 50% increase, implying there's a lot of protein binding to the surface. And this is, this is expected because there is no peg on the surface. Moreover, if you look at these fractograms, you don't need to calculate this shape factor to see that something is going on. The peak shown in red is much broader after incubation with the human plasma, signifying that there is some binding to the surface. So let me look at AF4 now to assess different manufacturing processes. So our client came to us with two different samples of a pegylated liposomal doxorubicin. So we subjected this to AF4 analysis, so process 1 and a process 2. For process 1, the results are shown here for AF4 moles and DLS. We can calculate the shape factor. We can see they're roughly the same before and after, so minimal protein binding to the surface. We also looked at the collection of these peaks to look at drug recovery, about 60% in this case. We did other analysis to look at the free drug, 8% in this case, as well as lipid peg content. What's interesting here is we did cryo-TM on these samples, and an example is shown here. These are around 66 nanometers with an aspect ratio of roughly 1, so they're nearly spherical. What's interesting, though, is because you have doxorubicin crystallized in the interior, you can look at the crystal shape morphology, and we calculated the linear and the circular drug shape morphologies from the TM micrographs. And based off of process 1, we're seeing about 86% with a linear crystal shape. If we look at process 2 and subject to the same type of analysis, AF4 with moles and AF4 DLS, we see that the shape factor actually increases. We get higher drug recovery for this sample, but the same free drug and same lipid amount in the formulation. But looking at the cryo-TM, it's interesting because the size is larger now, still spherical with an aspect ratio of 1, but now with the drug crystal shape, we're seeing almost an even mix of linear versus circular. So we have the linear here and a circular drug shape morphology. And actually, because of this increased amount of circular drug, this explains why the shape factor increases relative to the previous process, because you have more of your mass distribution now to the surface of the liposome as opposed to the center. So we're now looking at AF4 as a way to look at possibly crystal shape morphology in formulations. So let us look at AF4 now as applies to LNPs. Like I mentioned earlier, LNPs are in the limelight right now due to the COVID vaccine. So we want to look at AF4 to analyze these particles. And we did, and it works obviously. So we developed a method to separate LNPs, and we've collected each of the fractions coming off of the AF4. And we did offline analysis to look at the lipid content as well as mRNA concentration as a function of size for our clients. We also can do protein binding assessment with LNPs, similar to what I showed you before with the doxo, onivide, and ambisome. So we took these LNPs, incubated with human plasma, ran it through AF4, calculate the shape factor, and they're roughly the same, implying that there is no protein binding to the surface. Also, the shape factor gives you an idea of the mass distribution. So this tells you there's something in the interior of the LNP because the shape factor is roughly 0.77. What we also did in this case, we collected fractions to look at zeta potential as well as particle concentration as a function of size. So there's a lot you can do with AF4 in terms of separating and then collecting fractions and doing offline analysis. And I showed you several ways of what we do with offline collected samples in terms of analysis. And just one more application of the AF4 methodology. We wanted to assess two different processes for LNPs. Process A was a physical mix of 5 LNPs. So they had 5 different mRNAs. They formulated each of these and then just physically mixed the 5. Process B is they took the 5 mRNAs and formulated that into just 1 LNP. So obviously, if you think about this, you kind of know the results. The results are shown here. So for process A, which is a physical mix, we collected each of the fractions coming off AF4. We looked at particle count, size, zeta potential, and mRNA concentration, as well as calculate the shape factor. We did the same thing for process B, and the results are shown here. Now, if you think about it, process B should give you a more uniform sample, right, as opposed to physically mixing 5 separate samples, and the AF4 results show that. So just another extension of how AF4 can be used to characterize LNPs as well as look at manufacturing processes. So let me switch gears now and kind of leave you with, I basically showed you what we know, how to do stuff. I want now to leave you what we still need to know. So everything I described so far are ensemble methods that are averages. They give you average or bulk properties. So for example, if you look at a batch mode DLS measurement, it gives you the overall size of the sample, but not how many nanoparticles are of a certain size. Now, of course, you can hook up DLS to an AF4 or any other suitable separation technique, and you can get the size of a fractionated sample. If we look at total drug loading, it gives you the total amount of drug. It doesn't tell you how much drug is distributed over a size range. Again, we're kind of answering that question with AF4, DLS, and collecting fractions coming off and doing offline reverse phase HPLC. But what we can answer is if you take the case of drug-loaded liposomes, these techniques I described cannot measure how many liposomes have drug and how many are empty. And moreover, if they are loaded with drug, to what extent is the drug loading? For this type of measurement, or to answer these questions, you really need to move more towards advanced analytical techniques. And really, you need methods that are based on a per particle basis. So we actually are assessing particle concentration. We have in our lab a DynaPro and a Malvern Ultra, which are based on light scattering. We can do resistive pulse sensing with these 2 instruments, and we also can do single particle counting ICP-MS. Each of these have their caveats, their limitations, and advantages. And we can use this to assess particle concentration, right, on a per particle basis. And this is more for reference that you can actually do it. So we're looking at particle count for Doxil, Sun Pharma, and Nanovid. And we can get the particles per mil for each of these using one of our resistive pulse sensing instruments. So it's a good step looking at a per particle characterization, but how do we actually look at drug loading for these particles? For that, we're actually assessing new technologies, and we're working with 2 different groups to look at this. The first one comes way of Imperial College, where they developed this SPARTA, or single particle automated Raman trapping analysis, which can actually look at size, composition, and functionalization without any kind of labeling at all. And this is done on a per particle basis. We're working with them now, and we actually submitted to them Doxil and Onivide. And with the following questions, what is resolving power in terms of a mixture of different size particles? Can you do this in plasma? Can you tell me how many samples in Doxil are empty or drug loaded? And also to assess the surface coverage of the PEG on the surface. We have preliminary data from them suggesting that there's very uniform loading of Doxil, and there's more variation of drug loading in the Onivide formulation. We also established a second collaboration with another group. In this case, it's Oxford HiQ. They're using optical microcavities as well as Raman scattering and refractive index sensing to look at, on a per particle basis, the particle size, drug loading, and drug unloading profiles. We submitted to them, again, the same 2 samples, Doxil and Onivide, with similar types of questions. They were actually able to measure the distribution of Doxil and Onivide in terms of their drug distribution, and they recently published this as an FAQ or application note on their website, whereby showing similar results that I showed you on the earlier slide. So this is actually a very good step in really, picking apart your nanomaterial, drug loading questions, which normally we cannot answer right now. With that, I'll stop here, and I want to acknowledge the whole NCL team for their dedication to the field. And finally, I'd like to thank you all for your attention. Contact information should you want to contact me, and I'll leave it there and answer any questions. Thank you.
Thank you, Jeff. Thanks for the great presentation. On to the questions. So I'll start off with the first question. So if companies were to, engage with NCL or academic labs to leverage, as you mentioned, the sort of the assay cascade from a physical chemical characterization or in vitro and in vivo characterization testing, what is sort of the best way for them to sort of work with you?
So the best way is to go to our website, and you'll see on our website basically ways to collaborate with us. So there's different ways. The first is through the traditional white paper process, whereby you fill out a white paper. It has different sections contained within it. Namely, you need to demonstrate efficacy in a biological system. You need to have a comparison with the standard care, and then have good CMC or characterization on your particle. Once it goes through the white paper process, there's a part 1 and a part 2. If it passes through that, then the assay cascade is free. So that's one way of doing it. We also have C creators, where you pay us to do specific assays. And finally, we have TSAs, where we have certain assays that are out that you can pay for. These are typically, drug loading, drug release, and plasma. So those are the 3 mechanisms. Most people go through the assay cascade. So if you have a concept that's mature enough, that's the best way to go, because it is a free service to you. And everything is outlined on our webpage. And if you can't find it or have further questions, feel free to contact me.
Great. No, thank you for explaining that, Jeff. I think that information was really helpful. So we'll take a look at more questions. So what do you see as a trend in terms of the physical chemical characterization requests that you typically get at NCL in terms of comparing liposomal versus polymeric versus metallic nanoparticles?
Yeah, so I think, regardless of what platform you're using, I think the CQAs or the type of characterization that we're asked are similar across the board from a top level point of view. So typically characterization, most people will have size and zeta, right? That's something most people do on their own, and they can do that. And usually most people have drug loading, at least total amount. So where we come in is we try to look at, one, to validate their results and their methodology by looking at size, zeta, and drug loading. But we can also further look at, free versus encapsulated drug loading. We can look at release kinetics in plasma, for example. We can do a lot of compositional analysis for them, which a lot of companies don't do, or a lot of clients don't do at all. So by this, I mean the different lipids in the liposome, or the different polymers in the polymeric micelle. We can do a lot of that kind of stuff. So it's more, I guess, exhaustive characterization for that project. Now, it really depends on the project itself and the indication and what they need help with. And we always try to structure a research plan with our client to best characterize the relevant material, right? The relevant stuff that you need to characterize that FDA may ask you about when you go to file an IND.
Thanks, Jeff. So we have a question from the audience. So from that slide when you showed the 5 mRNAs, so when you had the 5 mRNAs formulated together, did that yield better particles than when you had the separate formulated lipid nanoparticles when they were mixed together?
So so this is from our client, right? I don't know how they actually made them and things like that, but they did submit to us in process A and process B that I referred to on the slide. So based off of our AF4 methodology, I would say when you have the 5 mRNAs that are first mixed together and then formulated into a single LNP, you get a single uniform size and a better distribution in that case. Now, of course, I have to also caveat that and say it depends on the application, right? There's always the other side of the board, right? It's easier to make 5 single, right? And then mix and match depending on the specialized therapy versus mixing 5 together and then making 1, right? So there's always that trade-off, right, of what you need versus what's easier to characterize. Does that make sense?
Yep. And so, yeah, because clearly it may be easier in terms of when you formulate it, but then, as you mentioned, in terms of the characterization, you have to take that into account, especially if there's differences between each of the different formulations as it pertains to the analytics. So
And also from a personalized medicine point of view, right? Some patients may only need 2 of the 5, right? If you have one that has all 5, well, you have to go back and reformulate, right? Whereas if you have individual, you can just mix the 2 relevant ones. So there's always that trade-off, right, from both sides.
Yep, and then also question 2 would be, no, not question for you, but also question in general is like, do you need to have all of those 5 in terms of showing it based on the efficacy, right? So maybe they ask you to look at combinations of them, of 4, and so you'd have to show that you do need all 5 together. So
exactly, yeah. Assuming that's been already done in the R&D stage where they realize that these 5 work for whatever indication, yeah, you have to prove that.
And since we're on that slide, talk a little bit more about the sort of the shape factor and what that kind of tells you as it relates to that comparison there.
Yeah, so here, so obviously if you have a mixture of a physical mix, right? Again, you're looking at an ensemble, the bulk properties of that sample, whereas when you have 5 MRIs in one, right, you have a more uniform. So I think that's where that comes out. The shape factor here, again, you're trying to look at the mass distribution in this sense. So I would expect for LNP for that number to be lower, more like 0.7-ish if it's filled with something. In this case, you're seeing that for process B, right? So I would expect that. But process A, because you have a physical mix of all 5, it might be a little harder to kind of discern what that shape factor means. I think in this case, you'd have to run each of the 5, see what their shape factor is, see if they're the same or different, and see how that influences the overall shape factor on the actual mixture of all of them.
And when you compare each of the shape factors, that wasn't done though, right? In terms of each of the individual?
Yeah, in this case, no, because we didn't have access. Our client didn't give us the 5 individual LNPs. It'd rather just a mixture of the physical mix of them.
Okay.
Yeah, but ideally, yeah, you would run each of the 5 separate, do the type of the analysis, and then when you do the physical mix, then do the same type of analysis to see what you get.
Another question on AF4 for the audience. When would one need to implement the A4 methodology in the lab? Sort of a more general question.
Yeah, that's a really good question. So part of our pre-screen, when samples are accepted at NCO, it goes through a pre-screen which consists of DLS, batch mode that is, zeta potential, and drug loading. So typically batch mode DLS, at least for me, I use that as a way to basically compare our results with our client results and make sure nothing happened to the sample in transit, and just to make sure we're on the same page with the sample. Now, I like to do AF4 almost immediately because I like to do AF4 with DLS hooked up. So you have AF4 or flow mode DLS, because it gives you a better indication of the size distribution on a fractionated sample, right? You get more information out of it as opposed to batch mode DLS. Batch mode DLS, I mean, if you're familiar with it, it has a caveat, 3 to 5 times different in size for it to resolve it. If you have something that's 110 versus 120, you can't tell if you have that in a batch mode measurement. But if you hook it up to some sort of separation, let's say AF4, you can start to discern those different size populations better. So to answer your question, I like to do AF4 almost immediately because, one, you get size distribution. And as I showed you, you can look at drug distribution as well as drug stability pretty much in 2 runs, right? One with and one without plasma incubation. It's something we almost always do on most of our particles that come in at the NCL.
Yes. I think that that does it for the questions. So again, thank you very much for this presentation today. These slides and actually this recording will be available to the public. Thank you very much today, and thank you everybody for attending this webinar, and look for other webinars from this working group as well. So thank you very much.
Thank you.